But there are some types of the species of E. coli, particularly O115. These ones can produce toxins. And you will see in the PowerPoint that there are some heat-labile, heat-sensitive toxins, two types of the toxins. So it's interchangeable between enterotoxigenic and enteropathogenic. Okay? This is one of the questions I received. Now, I will briefly address. I have to stand here, sorry.
Oh, you're going to stand there?
Well, I don't know because they want to hear. No, it's okay. It's okay.
All
right. Now, what happened? Why is not showing the? Oh, here's this. Now, one of the topics I like to address it briefly. You're going to study it again in details, and I tried to make it different than previous ones for two reasons: to see how good you like this or the previous ones. And there is a lot of names in relation to the disease and organism. So I said you may get confused when I talk about it without PowerPoints. But some of these diseases mainly transmitted by vectors. So clinically, we don't see it much. Some of this. So we don't see it much. But unfortunately, when you sit on exams with UCMLE or others, usually they ask about vectors, way the disease transmit, right? Mainly about this. So bear in mind that I'm not going, if you see any more clinical information, forget about it. Don't study it much. But I need to ensure that you are familiar with the name of the organisms plus what is the main disease associated with. Okay? Right. Now, this is a group called spirochetes, Spirochete aeris, with two families, big families, spirochetes, Spirochaete with species like Treponema and Borreliosis. These are important two species, and Leptospira, and the genus Leptospirosis and Leptospira. Now, the main thing in this group, as I will mention, that the Treponema pallidum. Treponema pallidum, one of the sexually transmitted disease. It has, you will study it more detail later on, but it has three stages: primary stage, secondary stage, third stage. Now, why I'm addressing this as a basic information? Because in the first stage, usually characterized by formation of what we call it chancre. So don't forget this word, chancre, right? This is one of the soft ulcer, painless. It is without pain. So what happened in the syphilis stages usually disappear after a week or 2 or 3, and then it moves to the secondary stage, third stage. Now, if you can recognize it from the clinical point of view in the first stage, you can treat it. But otherwise, usually it's very hard to treat syphilis if it comes to secondary or third stage. Basically, this is a message I like you to why chancre is important, because there is another sexually transmitted disease, not sexual. There is another disease caused by an organism called Haemophilus ducreyi. These also produce a sort of a chancre. We call it chancroid. When you say oid at the end, what is oid at the end? Like we say alkaloid. So it's chancre, and similar to the chancre, chancroid. These are same thing, is a hard ulcers or sores with you don't feel painless. You don't feel any pain with it. But the difference between Haemophilus ducreyi and the Treponema pallidum, that Haemophilus ducreyi with the formation of chancre can be transmitted by different ways other than sexual. But treponema usually sexual. So from the clinical point of view, if you see a chancre, you will look at the either soft or hard, and then you can tell, because it's not fair for someone infected with Haemophilus ducrei, not sexually transmitted disease, and you confused with the treponema pallidum. So this is a message for treponema pallidum. Now, the organism never grow in vitro. You have to do serology. So there are several serological tests we usually use to diagnose treponema pallidum or syphilis. Because the difficulty is there is two types of tests, specific and non-specific. So if you say specific, means you use the same organism in the test, the original organism. And the non-specific, you use something, an antigen similar to the microorganism itself. Okay, this is the difference between specific and non-specific. Now, usually when we diagnose syphilis, we do non-specific test because it's expensive to carry out a specific test for all patients diagnosed with syphilis. So we carry out what you call it non-specific test, and you will see it in the PowerPoints. And then if it's positive, we confirm it by a specific test. So this is a message for treponema pallidum. Now, what is the general characteristics of these spirochetes? Is a gram-negative and actually extremely thin and very long. It's from the name, spirochetes. So what it has here, it has what we call it endoflagella. You know, flagella in bacteria responsible for motility. This one, there is no flagella, but it is a spiral form, right? So we call it, it has what you call it helical cells with tapered ends, and this is what you call it periplasmic flagella. We call it usually endoflagella. So these are the structures of the spirochetes itself. This is how it looks like. I don't want you to memorize that, but the message here is that the presence of the endoflagella as spiral form in case of spirochetes. This is the message from this slide. And at the same time, although you cannot grow this organism in vitro, there is no culture media, most of the tests related to serology, but it has cell wall, cell membrane, outer envelope, and so on. Right. There are several names of genus and the species and some of the diseases. You have to memorize some of these organisms, and I'm sure when you go into the blog, you will hear about it again. So the three different species with a group of spirochetes. One is the treponema, different treponema, different diseases. Although we don't see other diseases like pigeon, Lewis, and Penta, but you have to be familiar with. You may not see it frequently in the kingdom or in the Middle East countries, but usually you may see syphilis. Even syphilis now, you don't see it much in the region, but it's there anyway. So you can see that different species, treponema pallidum, and different species in relation to the pallidum. So pertenue, endomycium, and the pallidum itself. Another disease or another genus called proli. This proli, different species. Again, unfortunately, you have to memorize it, but I'm trying to, this PowerPoints, it was almost 50 slides. I reduced it to about 20 or so, just for sake of concentrating on just the name, the disease, and a little bit some information in related to the organism itself. So we have Burgdorferi, it calls what you call it Lyme disease. This is one of the arthritis disease. You will hear it more in the blog. we call it Lyme disease. And is due to a microorganism of the liver. There's another species called recurrence. This one causes what we call it epidemic relapsing fever. And there are other species can cause endemic relapsing fever. And then we have another genus called Leptospira enterogens, and this is called leptospirosis or Wells disease. So these are the most 3 genus in the spirochetes group, right? Now we talk about Treponema. Simply, the Treponema, there are non-venereal Treponema disease. We don't see it much, but this is usually in other countries. We don't have to mention, but one of the diseases, we call it non-venereal disease, is not a sexually transmitted infection. BJ, Use, and Benta, and this is a primitive tropical and subtropical regions. Simply one slide for each, because I don't want to more details, that the Bijan, one of the endemic syphilis, usually start with an oral lesions, and then it becomes late as granulomatous of the skin, bones, and other. Again, I'm not concerned about clinical. I don't want you to know that, but this is simple one PowerPoint on the disease itself. But what I like you to know here that this one transmitted from person to person by contaminated eating utilities. So it's nothing to do with venereal disease, nothing to do with sexually transmitted. So we see diseases, but we call it non-venereal disease, found in different parts of the world. Use, now this is again one of the granulomatous disease. Start with the skin lesions, and I took the pictures. I have some pictures, but to see it, transmitted again by direct contact with lesions contaminated with the microorganism itself. I don't want to do that, and I will take out these PowerPoints, but this is how it looks like use. As I said, we don't see much use in the area here. Penta, one of the non-venereal disease, primarily restricted to the skin, about a few weeks as an incubation period. It has some clinical manifestation as initial or first or secondary or late, like we see in Treponema, again transmitted by direct contact with skin lesion. And this is how it looks like with the disease. So these are the 3. I will go back again. This is the, so what I like you to know from this slide, that spirochetes gram-negative, that extremely thin, it has what you call it endoflagell. This is more, for me, is more than enough. And there is no culture media. So most likely we do by serology. And then this is how it looks like, typical bacteria. However, you cannot culture it in vitro. You have to do serology for it. Unfortunately, it has some few genus and species, and with each species, it causes different disease. So we see syphilis, one of the sexually transmitted disease. Bijan, Use, Penta can be transmitted by direct contact from person to person. Now, these are the 3 common organisms. Now, if we go now to venereal disease, the syphilis, this is a sexually transmitted disease. They change the name, and I should change this in PowerPoint. Now you will see it in the recent textbook, sexually transmitted infection, not STD. Now they call it STI, sexually transmitted infection. You need to know that this disease can transmit it congenitally from the mother to the baby, as you will see. This is how we see it under the dark field microscope, the Treponema pallidum. And so what is the general characteristic? So this is too thin to see by the light microscope. whether you stain it with what we call a GEMS stain or gram stain. Motile can be seen with, we use what we call a dark field microscope, and then stain with some antibodies labeled with fluorescent dye. So the main detection of Treponema in the lab, you have to have fluorescent microscope. So we take the antibodies and then we add labeled antigen. If the antibody is similar to the antigens, right? And that's the fluorescence will appear. So it gives you a positive reaction. One of the intracellular organisms, you cannot grow it in vitro and do not survive outside the host. It must be transmitted from as a sexual, and you have to take care when you take a specimen transmitted to the lab for testing it. What is the epidemiology? Transmitted directly by sexual contact or from mother to fetus as a congenital, not highly contagious, and it has a long incubation period during which time host is non-infectious. And this is important from the clinical point of view. What I'd like you to know in this stage, the virulence factor, as we discuss it with other microorganisms. So you know the genus, the species, and then the pathogenicity. So what makes these bacteria pathogen? So you have an outer membrane for these bacteria that can promote adherence. It has what you call it hyaluronidase, and I'm sure you heard about hyaluronidase enzyme in different microorganisms, and these facilitate penetrations. That's why sometimes we call it spreading factors, because it allows bacteria or to facilitate the bacteria to penetrate. It has antiphagocytic coating of fibronectin, and then the tissue destructions due to these bacteria primarily related to host immune response. So these are the pathogenicity of the Treponema pallidum. I don't want to go into details. You are not supposed to know all this information, but my message here is a good information to hear or to read, but my message that it has what you call it formation of chancre. So the first stage of the Treponema infection or syphilis, formation of the chancre. And the reason I concentrate in that, I'm not sure if I give you the other species, Haemophilus ducrei, but Haemophilus ducrei form what you call it a chancroid. So usually from the clinical point of view, you may get confused of the source of the infection, whether it's a chancre or a chancroid. This is the message from these slides. Now we go to the secondary. I have to put it here as a PowerPoints, but this is actually appears about 10 weeks after the primary lesion, and then usually disseminated, mucocontinuous rash, and usually secondary lesions of the skin and the mucous membrane is really highly contagious. Now it causes a third stage or late stage, and for the first 4 years of life or infection, we call it early latent disease, or maybe after that, we call it late latent disease. Now, what is important also to know that the syphilis or Treponema pallidum can be transmitted transplacental infection. We call it congenital syphilis. So you may see in your practice later on what you call it congenital syphilis due to transmission of the organism from mother to the fetus itself. Prevention, I don't want to go into detail, but again, this is one of the organisms still until now sensitive to penicillin. But the message here that it can be cured if you manage to treat it in the early stage or the first stage of the infection itself. Once it goes to the second, third stage, it's very hard to treat. Now, because this is a micro course, you may have some tests in the lab, but these are the diagnostic. These are important to probably to remember. Either we're using the microscope by dark field or direct fluorescent, as I said, antibody staining. Culture is not available. And then, as I said, specific, non-specific. So the specific, we don't use the organism. We do something, an antigen similar to the organism. We call it non-treponema test. So the most common non-treponema test, and you should know it by now, the VDRL. A common test is a venereal disease research lab. Some other countries, they have what you call it RPR, rapid plasma region, or originally it was called Weatherman test. These are the tests. We still use it until now for diagnosis of the syphilis, but using what you call it non-treponema antigens or test. If you use the organism, because to culture the organism is very hard. You have to grow it in animal and then to collect the organism and use it for the test. Very expensive, not available. So usually we screen by non-treponema test, and what is positive, we do the treponema test. One of the important or two important tests, what we call it FTA, fluorescent treponema antibody absorption from the name, and microagglutination test of treponema pallidum. So serological test in case of the treponema pallidum. Any questions? So the message now for treponema, different genus, the one non-venereal, venereal, the non-venereal visual penta-use. This is transmitted by contact from person to person. Venereal disease, mainly the syphilis, and these have 3 stages. It can be easily cured from the first stage, and usually we diagnose it by serology. So this is a message for the spirochetes. When you go to the 3rd year, you will hear more of the clinical, and later on, you will see probably some of these infections in hospitals. Proli, this is, we usually use, like we do with treponema, we don't use Cram stain here, we call it Gemsa stain. So remember the stain. Gemsa stain, we use it usually for the spirochetes with the group. See, this is one of the organisms, we see it under the microscope or under the phase contrast microscope, how it looks like. Now, I don't want you to memorize that. This is not to keep in mind, but because in the future, if you see it, you will practice how you see it. But the message here that we have different Borrelia species. One Borrelia recurrence, we cause what you call it relapsing fever, and this is one of the reservoir human, and the other Borrelia species in relation to relapsing fever and the Lyme disease, and this is usually comes in your USMLE exam, and these are transmitted by ticks or by animal or zoonotic infection. And these usually transmitted by body lice and other tick. So some of the tick, we call it soft tick and hard tick. So this is the message from this slide. But I don't want you to memorize the name of the tick itself. But I need to tell you that Borrelia recurrence, this is one that infect or present in human. It causes relapsing fever and transmitted by body lice, or lice, اللي هو الأمل بالعربي. Borrelia other species can cause what you call relapsing fever or Lyme disease. Lyme disease becomes now more common because this disease usually clinically it's sort of what you call it arthritis disease. So you remember when I was saying that most of the disease we hear about it, we may be find out what is a causative agent, or there is an agent that causes. So some sort of arthritis is due to an organism. We call it Borrelia burgdorferi, from the name who discovered the organism itself. Now, what is it? The relapsing fever, these are the arthropods vector associated with poor countries or poor places or poverty. And these either louse or lice-borne Borrelia disease that cause epidemic, and some others can cause endemic disease. So the message from the slide here that there is always a vector, there is always arthropods related to the infection itself. What is the pathogenicity? It is acute infection for a few days, and this is the incubation period, followed by what we call it recurring febrile episodes. And from the name, relapsing fever means fever comes for a few days, disappeared, and then again, and so on. So this is what you call it relapsing fever. In relation to epidemic and endemic, the main difference between the both is actually one transmitted by Lewis and one transmitted by tick as a vector of transmitting the disease itself. I concern mainly with Borrelia burgdorferi because this one, as I said, this is by different stages. You may see it later on in the third year, but start with unique skin lesions like leukemia, migraines, and with the general malaise. And this can be subsequently cause some neurological or direct involvement with the patient itself. And it ends up in the third stage that it ends up to arthritis. This is common the rash we see with erythema, migraines of the Lyme borrelii. Now, what is the epidemiology? As I said, you need to know the names come from a place in the state in 75. That's why we call it Lyme disease, in a place in Connecticut called the Lyme, transmitted, by, as I said, by hard body tick as a vector. This is the message I need you to know from this slides. The other Leptospira integrans, this is actually originally, a few years ago, it was named as icterohemorrhage. And from the name related to the hemorrhage, one of the obligate anaerobes, and characterized by what you call it hooked end, as you see it in the slides here. Now, what is the pathogenicity? It causes what we call it Wells disease. This is the direct invasive, and it replicates in the body, and characterized by acute febrile jaundice. And it causes what you call it immune complex glomerulonephritis. So this is it usually has two clinical stages. I don't want you to go into details, but you will study that later on in the block itself. What is important to know about Leptospira, that it is a zoonotic disease transmitted to human from animals, animal host, most likely in the United States, the common reservoir, usually rodent or rats, dogs, farm animals, and so on. So it depends. If you have this host, you may see Lyme disease. If you don't see the disease itself. So usually is a zoonotic infection. And usually what I call it, one of the disease related to if you deal with the animal or your animal handling. So this is the message for the spirochetes. I know it's a lot of names, but again, all I need you to remember that that these are the species. You will study it in details later on. 3 genus, important to remember: Treponemal, Borrelii, Leptospira. You should know even name of the disease itself, because you will come across it later on, putting in mind that these are transmitted by arthropods vectors. So if you don't have the vector, you're not going to have the disease. what is important in this PowerPoint that mainly with the syphilis, Treponema pallidum, and usually you're going to see congenital syphilis transmitted transplacental from mother to fetus itself. And you have the 3 stage, you will study it later on in details, and usually you cannot culture the organism, usually you have to do serology, okay? So it may be a lot of names, but these are the important points I like you to know it, at least in this PowerPoint. Now, let me go to the TB. Sorry. Yeah. Let me go to the TB. Again, just because I need you to a few points to stress on. It's okay?
Yeah, I'll just set it up.
Okay. Oh, yeah, I see. Now, in relation to TB, as I mentioned last time, one of the disease commonly in Middle East countries and in the Gulf, and we have a vaccine against TB, we call it BCG. As I mentioned last time, one of the live attenuated vaccine and from bovine strain of the TB. So what is TB? It is an obligate aerobes, and again, I minimize these PowerPoints to very few number of the PowerPoints itself. And it is a facultative intracellular pathogen. One of the slow growing bacteria. It takes so long to grow. That's why we initial diagnosis, we usually do stain. We call it acid-fast stain. So these acid-fast, we call it acid-fast bacilli because of the lipid-rich cell wall. I call it mycolic acid, if you remember last time. So it actually can resist decolourization by acid, and you probably see it in the lab. So you see the mycolic acids, and then under this with the peptidoglycan and cell wall as well. So we do the acid-fast stain to identify the microorganisms. Now, this is the structures, and I'm sure you know that from the basic. It contrasts between structures of gram-positive bacteria, gram-negative bacteria, as well as microbacteria. We usually, the counter stain we use either methylene blue, it comes blue background, or if we use malachite green, it comes as a green background. You need to remember the media. The name of the media, low stringency media. So there are some media, usually comes in OSPE exam or MCQs, that, for example, for diphtheria, you need to remember low-floral serum media. For TB, low stringency media. For Neisseria, if you remember, Neisseria gonorrhea, Neisseria meningitis, we use cyan multimedia. Same when we say about Staphylococcus aureus, epidermis, we use mannitol salt agar. So these are the special media. We use it for each group of the microorganisms. It's very important to know this information because from the in the lab, if you suspect a disease, we usually specimen, we grow it in a general media, and at the same time on a specific media. So we save time to identify the microorganism. Because if you grow the specimen on general media, you will get a lot of mixed microorganisms. And if you grow it in a specific media and you get a culture, growth of certain bacteria, you can tell. And other media, you need to remember the media used for cholera, TCBS, thiosulfate, citrate, sucrose, agar. So this is the different media specific for the bacteria cell. So this is the lowest stringency media. Now, as a medical student, you don't have to remember the composition of this media. I never ask about the composition of the media. I ask only about what is the specific media to grow certain microorganisms. So bear in mind that if you have, at least in my case, if you have PowerPoints, concentrate on the main points. I know sometimes you may ask me how much I concentrate on, but as I said, you talk about main general characteristics, the pathogenicity, and particularly what is the virulence factor related to, and then how you culture this organism, if there is a specific media, and that's more than enough to know about the microorganism. Because for you, there's a quite few number of organisms and disease itself. For me, the reason I put this PowerPoint is because I need you to know that this bacteria resistant to most of the chemical disinfectant like phenol. So if you're going to work in the hospital, when I give you lecture on infection control, use sterilization and septic disinfectant, you need to ensure that how can you get rid of the TB itself. So the organism actually resistant to most of the chemical disinfectant. Usually it can be destroyed by tincture iodine, and you have, if you're using ethanol, about 70 to 80%, it must be for longer time, right? So this organism is sensitive to formaldehyde and glutaldehyde. You will mention, I will explain to you later on, formaldehyde, glutaldehyde. Formaldehyde we use for sometimes for fumigations in hospitals. Glutaldehyde, usually we use it to sterilize, to remove most of the microbes from the endoscopes. Later on, you will see that. Now, the organism, if you work in a hospital, you need to know that if someone diagnosed with TB, it must be isolated. You cannot admit it to the hospital. So you have to isolate, or you put him in a negative pressure room. This is a key important information, and I like you to know it. Otherwise, I'm not interested in the other information, because if you diagnose, so what usually we do, if you suspect clinically someone with TB, you take a sputum, you send it to the lab, see if it's an acid fast or not. At the same time, you must prove that you initiate therapy. But at the same time, you have to prove that by culture. So the viability of this organism in the sputum, about few hours, as you can see, droplets, a source of infection, is about for a few days, and cultures, it takes a few months for the culture itself. So this is important information to know about this. So the antigenic structures here, due to presence of mycolic acid, so they have different cell wall antigens, and they have what you call it cytoplasmic antigens, as you can see from the PowerPoint. What is the virulence factor? Like we do with other microorganisms, there are few. I mentioned only 2 or 3. One called cold factor, C-O-R-D, cold factor. This is one of the glycolipid molecules and the cell wall of the TB. And serpentine growth. This is the way the TB can grow as filaments or as cold. So it can grow in what you call it close parallel arrangement, and it can be toxic to the leukocytes, and it has a role in formation of granulomatous disease in the case of the TB itself. Sulfonalipids, these are the one of the substances, very important virulence factor for TB, because it will prevent what you call it phagosome lysosome fusion, which is important for the intracellular survival. That's why TB survived in the macrophage, survived intracellularly, and that this presence of sulfolipids allow the TB to survive for a longer time. I'm sure you know from Darwin that one of the immunity due to the TB is actually related to what you call a delayed type hypersensitivity or type 4 hypersensitivity. This is now important to know that how it is spread by droplet infections. So you need to apply what you call it standard prevention control, wearing N95, and as I said, you put the patient in the negative pressure room. Now, what is Important to know as a basic information for TB, I'm not talking about clinical now. I took all the slides related to the clinical, but I need you to know that if someone exposed to TB, they may not know infection, some percentage get an infection. However, this infection about 10% can get active TB. However, 90% get a latent TB. This is very important to know because in hospitals, you will know that later on, we usually screen healthcare workers for what you call it latent TB. You may have TB, but there is no symptoms. So you may be a source of transmission. This is what we call it latent TB. They will never develop to active disease. In active TB, some can be treated, some can be untreated, and they can die for a few years also. So this is the message that there is an active, but some percentage, high percentage can become what you call latent TB. This is a source. And I remember one of the master students, he did a survey in one of the hospitals for the percentage or the rate of the latent TB, and to my surprise, it was really high. So I think later on, when you practice, you will ensure that you have to screen for what you call it latent TB. So latent TB, this is the one of the TB infection, no disease, no sick, no infectious, but they carry the microorganism. However, if you contrast it to active TB, and this is to treat or to prevent transmission, it has actually the disease itself, usually sick, usually infectious, and is actually not infectious if it's not pulmonary, usually. However, usually infectious is pulmonary because there is pulmonary and extrapulmonary in relation to the infection itself. What you should know about lab diagnosis, either you grow it in a medium or you do what you call it direct Zen-Nelson stain, and sometimes we use what you call it physical examination. This is how we see the sputum. So if we take a sputum and see it under the microscope, we look to, what time?
We have 15 minutes left.
We look to the number, how to interpret the sputum. If you stain with the TB, you don't have to memorize that because this is more of the lab issue. But at least for your information, if we see more than 10 bacilli per the feed, we put these 3 plus or 2 plus or 1 plus in relation to the number of bacilli in the sputum itself. And very clear and very accurate if you want to diagnose TB. Usually what we do when we take a sputum, 2 things, and this is a basic, I think, it's nothing to do with microbiology. Now you need to ensure that you deal with a sputum and not saliva, because if you ask your patient to give you a sputum, he may not be able to give a sputum, but he may give you saliva. So now you should know how to differentiate between sputum and saliva. So do you know the difference between sputum and saliva? How can you tell the specimen is a sputum and not saliva? So I will leave it to look at it. So this is one of the important steps before you do diagnosis, you have to ensure that you're dealing with a sputum and not with saliva. If you have a saliva, you will get negative result. And this is, but you have to have what you call it a sputum. So what we do with the sputum, we add with sodium chloride, incubate for a few minutes. You don't have to memorize that. But the reason we do that is to destroy any other microorganism. And so what we were going to see in the field, we're going to see TB. Because if you take a sputum, look under the microscope, do acid fast, you may have difficulty to see the microorganisms. So this is I don't want you to memorize the technique. You are not supposed so. But the concept that if you need to ensure first using the sputum, and then to get rid of whatever other microorganisms in the sputum itself by using sodium chloride. And then you look it under the microscope. And then you culture it was what you call it Lewiston-Jensen medium, LJ medium, incubated in CO2, and it takes so long for identify. Now, the difference between pathogenic TB and pathogenic mycobacterium, non-pathogenic mycobacterium, pathogenic mycobacterium takes so long to grow. Non-pathogenic mycobacterium usually take 1-2 days to grow in the media itself. So this has been difference between pathogenic and non-pathogenic. Now, I don't want to go into details for the treatment, but again, I usually stress, I'm not interested to know the drugs at the moment, but I'm interested to tell you that usually we see a lot of TB cases in Middle East countries related mainly to two things: patient compliance and resistant, because we don't have much new anti-tuberculosis drugs. So we start seeing now resistant to TB, and it takes so long to cure TB. That's why we call it related to patient compliance. Usually you see TB in poor countries or in developing countries. So people are unable to provide the drugs itself. So that's why diseases are common in this area. Right. Now, PCG, as I mentioned, I'm not going to go into detail. You had it in PPL, and you know the interpretation of the TB. What we call it, we do the tuberculin test. We call it Mantoux test, and I'm sure you studied that in one of the cases in BB. So how you interpret tuberculin test? This is how to interpret if the diameter of induration less than 5 or 6 millimeter, greater but less than 15 and more than 15. And this is how we interpret and what action you should take when you interpret the induration. I'm sure you discussed that in the PBL cases. As I said, it's related to type 4 hypersensitivity, and I'm sure you had this in immunology, but you need to know that sometimes you may get false negative tuberculin test. Usually false negative tuberculin test in case of miliary TB, disseminate TB, convulsions are from measles, other malignancy disease, or CMI, or inactive PPD, or improper injection. So this may be the cases due to false negative tuberculin test. False positive tuberculin test, if you have an infection of what we call it atypical microbacteria. We have now, I don't want you to study this in details, but now we have what you call it rapid methods to identify the tuberculosis. The reason I don't concentrate on that, because these machines not available in all hospitals, available only in certain hospitals. So we have what you call it Pactic machine, and this is fully automated, and you can identify TB within few days rather than you waiting weeks. But unfortunately, this is not available in all hospitals. But at least the message now that there is a new machines, you can identify this. And of course, we use what we call it PCR, and we do some serology with antibodies against TB. One non-medical information, DOTS therapy, as I mentioned last time, with the direct observer therapy. So if you follow your patient, ensure he took the drug on time, ensure availability of the drugs. WHO believes that you can cure TB within 3 months. So this is the message from the TB. How we apply it, when you apply it, depends on the country, depends on the availability and so on. But there's a way to ensure that you cure TB. I don't want to go much to multi-drug resistant TB, but this is a case. This is the actual case problems in Middle East countries, because we still have the same drugs I'm teaching for anti-tuberculosis 40 years ago is the same drugs I'm teaching now. We don't have any new drugs for the treatment of TB. So we start to see a lot of multi-drug resistant in TB. We see now extensive drug resistant TB. Finally, I need to ensure that you know some prevention methods. You wear mask, you give the vaccine, medical follow-up, isolate the patient in a negative pressure rooms, ventilation, natural sunlight, and then the radiation itself. And one of the questions I gave it in one of the exam in relation, where can you take sputum specimens? So I give them few options. And now, by now, if I ask you when or where you can take the sputum specimens, you should know where you will take the sputum specimens. You don't take it in the inside the hospital. You don't take it with a lot of people around you, and so on. So now you know when you take the sputums in case of someone with the TB. This is a message. So I hope it's clear now for the group of spirochetes and the TB itself. And I will put the slides for mycoplasma, chlamydia, and for the legionella. I will put it today in the PowerPoints. And you're going to have the exam in a week time, okay? Any questions? Again, if you have any question, email me or come to my office. If I'm available, I will respond to you immediately, no problem. Don't worry if I have someone inside the office or I have a meeting. Come to me and I will respond to you immediately, okay?
Thank you, Doctor.
You're welcome. So, inshallah, on Sunday, Abrar will give you a lecture. On Sunday, I think, and Monday. And then I will give you just briefly related to infection control, what's antiseptic, disinfectant, sterilization. Very basic, not much on the Thursday. And then you will have the exam, I think, on Monday. Yes, on Monday. Okay. Now, as per your request, I'm not going to put any question from next week lectures. Okay, that will be the last lectures you will have. And I put 2-3 MCQs, one on TB, one on the others. So you will have 1-2 MCQs for that. Okay, I'm not hiding anything. If you need to know whatever, tell me. If you want even to know the questions, I can give you the questions. No problem.
How many questions is the exam?
And I think so far I decided about 80 MCQs. 80, yes. In the final, maybe more than that. And it will be short answer as well in the final exam.
So for the midterm, only 8.
In the midterm, only 8. And only MCQs, no short answer questions. We're going to have Sunday LGD. I don't know if you heard about LGD or not, but we're going to have LGD with Dilip and Garwin. So if we have a case, a scenario, if you have any questions related to this, we can address it. If you have anything related to gram, we have one case on gram-positive, one case on gram-negative bacteria. And I try to include micro, pharmacology, and immunology. And then Abrar, he will give you LGD at the end of the week, next week, by himself, because he would like to discuss more of the pathology cases. Okay? Thank you very much.
Thank you, sir. So I'll just
Can
send
I
it.
take my PowerPoint?
Yeah, sure. Your USB, right? Yeah, I'll give you any of them.